second part of the innate defense system is a so-called interferon system, which are specifically existing in the vertebrate cells. So vertebrate cells, almost every cell type can produce interferon. So it's host specific and not virus specific, meaning that different species can secrete different forms of, slightly different forms of interferons, but they are not specific for certain viruses. They're a broad spectrum antiviral defense system. It can be produced by virtually any cell invertebrate animals, invertebrates. They are induced by virus infections such as the double-stranded RNA molecule, as well as which are signature of certain virus infections activated by intracellular bacteria. Interferon is a first line of defense against viruses before a specific immune response is mounted. Currently, there are 3 types of interferons known, which can be classified into type 1, type 2, and type 3. So the type 1 interferon are contain the so-called alpha and beta interferon. They are the main functioner in inducing cells to become resistant to virus infection. Interferon gamma is also called the type 2 interferon. Its main function is to modulate immune responses, but type 2 interferon also have its own antiviral activities. And for type 3 interferons, they're more recently discovered, and their function are still being studied. So the well-known functions of interferons are listed here. These are interferons. They are basically small proteins that can function as a signal between the cells. They can function to activate macrophages, natural killer cells, and other lymphocytes such as cytotoxic T lymphocyte. They can also modulate the synthesis of antibodies. Basically, they have functions to modulate both innate antiviral defense as well as adaptive immune responses. They can induce MHC class 1 molecule expression, which I'll talk about later. And type 2 interferons can induce expression of MHC class 2 antigen expression, as well as the antibody receptor. All this help the adaptive immune responses. Interferon also is a master regulator of the antiviral responses. They control the expression of hundreds of so-called interferon-stimulated genes, which are the main effectors of innate antiviral responses. But interferon also causes so-called side effects. Since they have been used as antiviral drugs in humans, for example, treating hepatitis C infection, they are known to cause side effects, mainly fever-like syndromes. So that really causes the unpleasant feelings of the patient, which is which limits its application as a general antiviral therapies. It also can inhibit cell growth. Basically, the interferon is trying to change the state of growth into a state of defense, so that it actually can slow down cell growth, which also can lead to certain side effects if you use this to treat human patients. So how does interferon works? As I mentioned, interferon are small proteins released by cells into extracellular environment, so that they can send a signal to neighboring cells. Here it shows you that type 1 and type 2 interferon, when they are released into the extracellular environment, they can find their receptors on the surface of the neighboring cells. When the interferon bind to their cognizant or specific receptors, for example, here, type 1 and type 2 interferon, they have different receptors. And this binding then triggers a so-called signal transduction cascade, meaning that a bunch of a molecular event happens downstream of this binding of interferon to its receptor, which is characterized mainly by a number of protein phosphorylation events. As you can see, the JAK protein kinase are involved in this downstream event, which eventually activate transcription factors such as 1-2 transcription factors, as well as IRF9 transcription factors downstream of type 1 interferon. And the STAT1 they can form homodimers, which are activated by the type 2 interferons. So-called transcription factors, when they are activated, they go into the nucleus of the cells and bind to their specific DNA target sequences. And this binding then drives the so-called transcription of or expression of this genes with a sequence that bind to this transcription factor. Eventually, a cohort of so-called ISGs, interferon-stimulated genes, are then expressed from activated cells. And as I just mentioned, there are hundreds of genes that are now being characterized as interferon-stimulated genes. As you can imagine, this is an army of defense against different viruses. And that really changed the cell state into an antiviral state. I'll give you one example of the interferon-stimulated genes. This ISG, or it's called a protein kinase R because it's dependent on RNA. So basically, it's an RNA-dependent protein kinase. So this ISG is expressed when the cell sends interferons. And when it is expressed, it first exists in an inactive monomer state. Basically, they are self-inactivated. However, when this protein sends double-stranded RNA, so in our cells, most normal RNA, they are single-stranded. But when the viral RNA trying to replicate itself, it often contains double-stranded stage of the replication cycle. And this double-stranded RNA really becomes a pattern of or a signature of viral infection. And this double-stranded RNA can bind with the protein kinase R, which then activates this protein kinase, cause it to dimerize. Basically, two molecules form one complex. PKR dimer can then actually phosphorylate and further activate itself. Further activated PKR kinase then can phosphorylate its substrate, the protein so-called EIF2 alpha. And this protein is actually responsible for starting the protein translation process. Basically, PKR phosphorylate EIF2 alpha and inactivate this protein translation initiation factor. As you can imagine, if you stop the initiation of protein translation, all cellular protein production will be basically shut down. So this is quite extreme procedure, so that the interferon basically shut down protein production in the whole cells to stop virus from replicating. That's really an effective way, basically, to completely block viral replication. As I mentioned very early on, so there's always an arms race between the viruses and the host. Such an effective way as we know, didn't lead the virus to go to extinction. So virus must have evolved countermeasures against it. Many different viruses actually evolve different ways to counteract this PKR's antiviral activity. For example, the real viruses have encoded a core protein that binds to double-stranded RNA. So, and this binding then actually prevents activating of PKR. Adenovirus, which is a common cold-causing vector virus, can actually encode a so-called VA1 protein, also prevent double-stranded RNA to bind to PKR. So it's the pox viruses. Basically, the virus have evolved a way to shield their double-stranded RNA from being sensed by PKR, thereby evading this antiviral responses.